Hello. We are officially known as Northern Guilford because that's the county district that we belong to, but we're from Mendenhall Middle School. We were the middle school that won out of our district. We're from Greensboro, North Carolina. I am Lenny Sue French.
I'm Clay Herring.
I'm Anna Freund.
I'm Bailey Weichel.
And I'm Alex Atwater.
The last name of her, part of her name was Facus, in case you missed that. Okay, Alex was our principal investigator on this, and we were doing the development of brine shrimp, which has been done many times. In fact, it was the very first scientific experiment ever done in space. It was done on the Friendship. They actually took some. And you can see our little shrimp entering the Endeavour. See them? Yeah, they didn't look like that when we put them on, but we'll show you what they did look like.
Okay, so basically, like it says up here, we believe that the brine shrimp in microgravity would have less of a gravity force pushing down on them. So therefore, they would grow larger. That's our main idea. We use the MDA type 2, and we use distilled water and brine shrimp crystals, and we use 5% NaCl in the water. We also fed them yeast extract. So from our research, we found that salt is the main indicator for when brine shrimp actually hatch and become cysts. So we did some research and found out what that point was. So that's why we used two different sides of the MDS to put them together.
In the space sample, we found it was more clouded and a slightly copper color. In the Earth sample, the fluid was clear and there was less volume. Observations. Space samples, many globular-like spheres clustered together, reddish tint, no detectable brine shrimp. In the Earth samples, there are clear fluid with white stripes, cysts, some intact, others broken open, no detectable brine shrimp.
These are the images from the Helium ion microscope. These are the space samples. The eggs seem to be broken or cracked. The cysts surrounded by globules. They seem attracted to the opening in the egg. This is the Earth sample. The two cysts that appear to be hooked together, one is more smooth on the exterior than the other. This is the cyst closer up on the helium ion microscope image. This is like where the two eggs were connected together. This is like the exterior of the eggs, and you can see they're somewhat smooth. We thought it kind of looked like the surface of the moon. Our conclusion was our experiment results do not reflect what we thought was going to happen. However, it did reveal many items of interest to us, and so we're continuing our process of using the duplicate Earth experiments and further researching our microscope images with professionals in marine biology.
We also thought the globules could be the yeast that we put with the brine shrimp, and so that we could get another opinion, we sent it to the coast of North Carolina. Also, we thought that maybe the brine shrimp didn't hatch because the water was too salty, or the space was too small, or the eggs were just duds. And we didn't find any when we looked.
The early experiments showed that essentially they grow larger because of the lack of pressure on them because of microgravity, which makes perfect sense. It just didn't happen to ours. So we're trying to figure out why. Why did that not happen to ours? And so we decided we're going to do a bunch of other tests. So when we start up school next year, we're going to go ahead and we're going to test using the same salinity along with a lower salinity environment. And we're going to use eggs from the same batch because these eggs will stay in stasis for years and years as long as they're dry. So we're going to use eggs from the same batch. Obviously, I wish I could send some back up on a shuttle, but that's not going to happen. And we'll use one with the same salinity and see if it was just a fluke that it didn't happen the first time. Then we'll use less salt to see if we can answer that question. We're also going to test dyeing the yeast. We saw a bunch of experiments when we started researching that you can dye yeast like with a special red dye. And so if you dye them, then we would know, okay, if the globules in the image are red, those are in fact the yeast globules that we're seeing. We also thought that we could test using a standard 10 milliliter test tube, use the same salinity, use the lower salinity. One of the issues we had was the MDA was so tiny that even if brine shrimp did grow in there, when you tried to get them out of there, you would have broken them up into how many ever pieces. And of course, we weren't the only ones that did that. You know, ITA did it down there, and then we had to re-get them out up here. So we thought if we tried them in a bigger area, maybe we'd get something. And last of all, we wanted to thank, of course, Guilford County Schools is what we belong to. NASA and their response to the SSEP program has been phenomenal. The crew of the STS-134, especially Mr. Greg Chamitoff, who turned the crank. The N is for NanoRex, who's working so closely with SSEP and NCE-SSE, who are also mentioned on here, and with Dr. Jeff and Dr. Harry to make sure that they get the kind of equipment they need. And when you see Dr. Jeff's thing in a little while, you'll see what I'm talking about. They are actually making MDAs for SSEP, meaning they're going to get a lot more space, a lot more individual time, attention. Your experiment's going to be able to do what you want it to do instead of having to rely on 19 other experiments. So I'm absolutely jazzed about that, and I think, you know, NanoRex deserves a big heads up for that. Thank you very much. Are there any questions?
So was there growth in the space?
Well, like you can see in the picture, we're speculating that it does look like they are it looks like there is some growth. But that's why we sent it off to some biologists to get their opinion on what they saw, what they can see.
Thank you.
Not brine shrimp. Like the eggs are opening, like it looks like they tried to, but no actual whole shrimp were found.
Thank you.
What were the benefits of your experiment?
Well, personally, I did not know anything about brine shrimp, so I learned what brine shrimp were to begin with, and I learned that they do not grow in Miss French's bedroom, so yeah.
Apparently not.
Thank you.
If you did get the brine shrimp to grow, what would you do with them?
In the initial proposal, I mentioned that you could use this for, if in the future, you know, long space flights, you could actually use this for food for other organisms that we would be able to eat, like many fish, cichlids, other things. Lots of predatory organisms eat the brine shrimp, so that would be one implication. And it also shows, if we had gotten the results that we wanted, how organisms develop overall, their structure, everything else.
Originally, if you didn't have any other ideas for experiments, like, the question didn't come out right. What were they? Did you have any other ideas?
Do we have any other ideas?
Oh, yeah. We had, was it 6?
Yeah, we put in 6 proposals.
There was everything from rock candy to single-cell organisms to pinto beans and E. coli. They're naming everything. I'm just saying it. We had a lot of them. Actually, brine shrimp was one of the last-minute things that came up. So. Yes.
What exactly are globules?
Globule is a generic term for any kind of sphere or a round object that's three-dimensional, but they usually hook together. And so in this case, like in the picture, you could see where they kind of all look like they're sticking together. In fact, we thought when we first saw them, right there. Nope. And there. We thought when we first saw them that one down there in the bottom kind of looked like a brine shrimp, but it looked like a brine shrimp attacked by globules. Okay? And so that's why we wanted to know what they were exactly.
Yes, I was wondering where you got access to your microscopes.
Access to the microscopes.
Yes.
That was actually the Joint School of Nanoscience and Nanotechnology. They're a local research college. It's actually a joint, so two colleges have actually bring both resources from all students that are going into those fields actually go there, and they.
And the ion microscope, the one that took these pictures, that's a $2.5 million microscope. There are 4 of them in the United States, and we are lucky to have one in Greensboro. So we just happen to be blessed by the gods. That's how that happened.
Okay, thank you.
All right, thanks again for an excellent presentation.